Hey folks, welcome back for part two of neuron structure in the nervous system, also neuron function, I guess. So we finished here by talking about how an action potential is generated sort of in broad strokes. So now on to a little bit more nitty gritty. Um, and for some reason my link is not moving. There we go. So basically what we've got here, I called it the depolarization, and that's really what's referred to all the time. But if you were to look at an action potential as it's moving down an axon, so perhaps, as it's moving down the axon, there's a graph that sort of represents the whole thing. And I know it kind of looks like a PQRST wave from your like a cardiogram, but electrocardiogram, but it's a little bit different here. So we've got our resting potential here. So when your neuron isn't doing anything, it's just sort of hanging out. That's the membrane potential, the electrical charge across the membrane, on the inside in particular. When the threshold potential is reached, immediately what's going to happen is voltage-gated sodium channels are going to open up and sodium is going to rush in. That's going to begin the process of making the inside slightly more positive than the outside, which is exactly the opposite of how it is during resting potential. So that's where we get to that threshold, and the cell body decides to open up those voltage-gated channels. It doesn't decide; it's not sentient, but it gets enough input from its dendrites to open up those voltage-gated channels. Sodium rushes in. Then sodium continues rushing in. Potassium can't rush out just yet, but it's going to be able to complete this whole thing. So the depolarization phase, that sodium's just sort of allowed to freely rush in. And then we're going to shut the sodium channels so that the sodium can't come back out, and we're going to allow potassium to leave. And now what ends up happening is we're going to start seeing that we've got a more positive inside than outside. So we're going to reverse those charges, and I'll show you that in a minute. And then the last thing that we've got going on, move myself out of the way, the last thing that we've got going on is sort of this phase here, where sodium is allowed to rush out, potassium is allowed to rush out, sodium's not allowed to rush in, and we come back to our resting potential. So we've got our threshold potential, we have our action potential, which once reached, we're going to actually start pumping the sodium back out. Sodium's not going to be allowed to rush in anymore, and eventually potassium won't be allowed to rush out anymore either, and we're going to have this thing called the sodium potassium pump that's going to pump all the sodium back out and the potassium back in, and that's going to restore us to resting potential. But we do have a little undershoot phase. It's sort of a refractory period here where we're getting the sodium back out, the potassium back in, and we get back into our resting potential, which is the time between action potentials on the axon. So another way to look at this, we've got our generalized neuron here. Here's the axon helix, and the cell body is going to reach threshold potential. When it reaches threshold potential, sodium is going to be allowed to rush in. As the sodium is being allowed to rush in, the change in charge, making that inside slightly more positive than the outside, that's going to cause voltage-gated channels down here to open, which is going to cause voltage-gated channels down here to open, which is going to cause voltage-gated channels down here to open. That allows this action potential to continue moving down the axon. Just after that happens, the sodium is going to be allowed to rush out, which furthers the charge differential. It really flips that charge. And then we start going back to resting potential by pumping the sodium back out, the potassium back in, and going back to our resting potential. So you can almost think of it as the cell body opens these voltage-gated channels once the threshold potential is met. And that starts a chain reaction that literally just allows, like dominoes falling over. Um, Once sodium starts rushing in here, it's going to be allowed to rush in here and here and here and here and here and here all the way down. Potassium's going to rush out all the way just behind that, and then right behind that potassium rushing out, we're going to be pumping the sodium back in. We're going to be pumping the potassium, sorry, the potassium back in, the sodium back out with the sodium potassium pump, and that is going to restore our resting potential. When we get to the end, that's where we're going to communicate with the next neuron in line. And one thing that's important to talk about here is this idea called saltatory conduction. Some of your neurons are really, really long. They run from the base of your spinal cord, in your lower back lumbar vertebrae, all the way down your legs to your big toe. I mean, that's like a meter for some of us. And in order for you to be able to send a message quickly enough, if you just for a second, I mean, think about moving your toe and move it. Like it's such a fast thing to get from your brain down to your toe. Um, the only way that's possible is by having this thing called the myelin sheath and saltatory conduction. So let me move myself out of the way here. So basically, we have these things called Schwann cells. Here's the nucleus of the Schwann cells here, and it will lay down a fatty layer called the myelin sheath. And it lays it down in such a way that there are these little tiny gaps. And these little tiny gaps are called nodes of Ranvier, which is it's one of my favorite names in all of biology. But these little nodes of Ranvier. And basically what happens is once the cell body has generated or it has received enough input to cause the opening of the sodium voltage gated channels here, all that has to happen is you end up doing the exact same thing we just talked about, but only in the places where the myelin sheath isn't. And so basically you're skipping large sections of having to open up all these voltage gated channels and shutting all these voltage gated channels, which is one, time saving, and two, energy saving. So that's how that's going to pass that along on longer neurons, on neurons that you use a lot, and on most of your interneurons where you have to be firing really rapidly to do things. And it's really important. Also, as you learn things, as you cement them in your brain, some neurons are myelinated in a better fashion than others. So the things that are like right there on the tip of your brain, you can pull those facts out anytime. Those tend to be sort of like myelinated pathways that if you can hang new learning on, it will actually help you if you can connect new learning to those well myelinated pathways, that will allow you to sort of learn the new information more quickly. Sort of like if you have learned one extra language, it's generally easier to learn more languages. Yeah. So this is a more close-up view of that. So basically, here we've got our generalized nerve cell again, our generalized neuron, and you can see that we just jump from one node of Ranvier to the other, and we only depolarize in that node of Ranvier, and then it can jump ahead to open the voltage-gated channels and continue doing it. It's much faster. So if you're looking at non-myelinated neurons, you're talking about somewhere between 0.2 and 2 meters per second. On myelinated, you're going 12 to 120 meters per second. That's way faster. So once we get to the end of the neuron, the action potential comes down. This is an axon terminal. That's where those neurotransmitters are stored. And here is the dendrite of the next neuron, or it could be an effector, like a muscle or a gland. So basically what's going to happen is you are going to have a nerve impulse come down. It's going to hit these vesicles that hold neurotransmitter, and the vesicles, sort of like if you think back to the Golgi apparatus, it just had a, it made phospholipid circles around whatever it is that's trying to ship out of the cell. That's the idea here. We just have a membrane around, which is a vesicle around the neurotransmitter, which is going to be released out into the synapse. And remember, I said the synapse is actually open space. It doesn't actually connect neuron to neuron physically. It connects through the passing of a neurotransmitter from one side of the synapse to the other side. And there's receptors that can make things happen either in the dendrites of the next neuron or in the effector that you're getting to. Important to realize, though, that generally what's happening is once the neurotransmitter gets across, that receptor isn't going to carry a charge all by itself across. Basically, what's going to happen is it's going to open up some ion channels. It's going to allow sodium to rush in and propagate the signal to the next thing. So we've got, this would be the presynaptic neuron, this would be the postsynaptic neuron or an effector, and you have a little view of that here. So this would be coming from this neuron, this would be the dendrite of this neuron here, or like I said, an effector like a muscular gland. Okay, so these are just some generalized, not generalized, some very specific actually neurotransmitters. And you probably heard of some of them. The ones that come up the most often that you might want to just sort of, you might want to just sort of have in there would be acetylcholine. That's the one that's probably going to be making you move your muscles. And then there are these biogenic amines like norepinephrine. That is sort of your fight or flight. Your fight or flight response is going to be controlled by that. When you get called on in class and you weren't paying attention and your heart starts to pound, it's probably because norepinephrine is being released in a whole bunch of your interneurons. So you get that feeling. Dopamine, that's going to be one that is it's like a pleasure neurotransmitter. People, like when you're eating lots of like big chocolate cake and that kind of stuff, you can be releasing lots of dopamine, doing things you really like. That's dopamine. Also, dopamine tends to be the one that that leads to addiction because lots of drugs sort of mimic the effects of dopamine. So your body slows down the production of dopamine, and then you have to take the drug to get back to feeling normal, and that is literally the definition of addiction. Serotonin, that's going to be an inhibitor for your central nervous system, which is going to sort of help regulate sleep cycles and things like that. And those are really, if you're looking to be scientifically literate, those are probably ones that are the most important to know. Let's see. These are just some sort of nerve layouts for different organisms. And you can see not everything has a brain. I think that's really the most important part of this slide. So we've got Hydra, which are these little tiny cnidarians. The C is silent, that's fun. That have a nerve net. They don't have a brain. Neither do sea stars. They have a nerve ring. And then we get into planaria, which live in ponds and things like that. Still no brain, just sort of nerve nets. Well, there's a really, really small rudimentary brain. And then we get into leeches, which are like segmented worms as well. They're getting close to brains. They've got these things called ganglia along the way. And then once we get to things like arthropods, we have a really small brain, we have some ganglia, we have a pretty sophisticated nervous system starting to get put together here. And then we'll just jump ahead to an animal where we've got a true brain, we've got a spinal cord, we've got lots of ganglia that all work together. So you can pause the video, take a look at this. It's sort of interesting to see where things are as far as nervous systems go. And let's see real quickly, probably don't have enough time. So I guess I'll make a third video that'll be very short about the different nervous systems of the human body. See you then.